Quarter 2020. It is my pleasure to introduce Dr. Roderick Melnik. Dr. Roderick Melnik is a full professor at Wilfrid Laurier University in Waterloo. He's a T1 Canada Research Chair in Mathematical Modeling. He's also affiliated with the University of Waterloo and the Center for Applied Mathematics, PCAM, in Europe. Dr. Melnik has received many awards, including prestigious fellowship in Italy, Spain, England, and Denmark. He has published over 300 research contributions in mathematical modeling for sciences and technology, including biomedicines and bioengineering. Professor Melnik serves on editorial boards and has been a guest editor of many international journals and book series. Please join me in welcoming Dr. Roderick Melnik.
Thank you very much, Hina, for the introduction. Thanks, Stephanie, and thanks other organizers who invited me here. It's a pleasure to be in Winnipeg at this new institute, relatively new institute. This is the title of my talk. So what I would like to bring your attention to is that when we are talking about biological applications and ecological applications, non-local models are essential. And what I'm going to talk about, I'm going to give you examples from 3 different areas. At the same time, they're interconnected areas, including biosocial dynamics, ecology, and brain studies. As any work of this type, of course, it's kind of reduction of many other projects that we are doing. However, on this particular projects, it's different parts of this work has been done with a number of people who you see here, including actually Hina before. So other people are based in Swadesh and Sanhita are based in Waterloo. So this is outline of my talk. So as I said, there will be 3 groups of examples from biosocial dynamics, ecological pattern modeling, and neurodegenerative diseases. And I will start from biosocial dynamics. Then many things that I'm going to give you as an example are going to be related to predator-prey model, which kind of on the border between biosocial dynamics, if you're talking about animal kingdom or human society, and ecological also models. Then I'll talk more about influence of stochastic effects before moving to ecosystems. And then we're going to talk about probably the most complex part of the talk, which is related to neurodegenerative diseases because it's directly related to brain studies. And actually, this is the area where not only classical equilibrium dynamics is important, but many things are driven by non-equilibrium processes. And as I put it in the abstract, if you care to read it, it's effectively life itself is a non-equilibrium, where all these non-local processes within that are playing quite a crucial role. And at the very end, for those of you who might be interested, I'm going to talk more about, the connection to more like machine learning, artificial intelligence, specifically physics informed neural networks. Okay, and then I'm going to complete. So, so first key concept, why basically we need this non-locality. So first of all, as you know, kind of traditional way of modeling biological systems have gone forever after COVID. Now we understand quite well that we cannot describe just biological systems because what we need to do, you need to take into account social effects. And what you see here, basically in the first line, biological changes can alter social behavior on the one hand. However, social behavior can also impact biological outcome. And this is very often, if not in most cases, are done in terms of non-local interactions, where effectively distance influences shape system dynamics, overall system dynamics of biosocial system. And more than that, even small, relatively small, depends on the scale you're considering, small social changes can trigger large-scale biological effects. And one example during COVID, as you may recall, was vaccination, for example. And if you account for that, then the very last part of my talk may become essential because what you may need to do, you may need to consider also or to bring the machinery of non-equilibrium dynamics. The other part, which you will see pretty much in a few minutes now, is that the dynamics of biosocial systems exhibit special temporal patterns. Those spatial temporal patterns are very often driven by non-local interaction and non-equilibrium processes that may create, as you know from non-equilibrium dynamics, emergent structures. I'm not going to talk about disease modeling or epidemiology, although we are doing that mostly with my European colleagues. The very last point that I would like to point out is effectively memory effects, where past states influence the current behavior of this biosocial system, can introduce also non-local temporal dynamics, where these history-dependent processes shape long-term dynamics of these biosocial systems. So if you like, this is key concept in biosocial dynamics, and by that slide, I was trying to convince you that non-local dynamics, it's not something that's an exception. It's rather a rule, which, and in many cases, as mathematician or in other areas that we are working in, we are trying to avoid because of the simplicity of the mathematical models we are using. However, we have to face it in many cases. Now, if you move to ecology, what is interesting here is that, very often we have to look for patterns. We have to look for patterns not only in terms of spatial distribution, but also very often in terms of spatio-temporal distributions. On this picture, you see basically ecological pattern formation, which well studied and well known in biology, specifically pattern in vegetation. So left you see hot spot pattern, then tiger burst pattern, and then cold spot patterns. And the thing is that many processes, if you take even this simple example, we are not going to animal kingdom, we are not going to human society. We are talking about just vegetation. I mean, think about dispersal. I mean, is it a local process? Of course, it is a non-local process. You cannot describe the process with pure diffusion. So non-local models already needed here. Now, you move on to animal kingdom. And good point here is that, as you see, many of those patterns that you observe in animal kingdom, in many ways remind us those patterns that you've just seen on previous slide, which means that some of those models that we are using for ecology in plants or for vegetation may be applied in some cases even in this type through the idea of these patterns, how to study patterns in this ecological model. Now, the thing is that we haven't talked about is that, of course, again, if you go to animal kingdom, what you see competition, intra-species competition and inter-species competitions. And these interactions and competitions could be very complex. Of course, what you see on the top left picture is predator-prey kind of dynamics when you have one prey, one predator, which is simple. But then you may have one prey, two predators. You may have multiple, two or more prey and one predator, and so on. So that complex interactions would require from you to model, to account in your models, coupling interactions. And again, the way to study that successfully, in our opinion, is through studying the patterns in dynamics. Now, what we've just been discussed, of course, is also true at the very elementary level, at the lower scale. So you go to cell scale, you go to, for example, the bacteria grows. Now, you may know that basically the interactions within the cells are actually non-local. And the way how it's done is through cellular protrusion. For example, Philopodia is an example of that. So if you like, it's a sensor mechanism within the cell that transfers the information not just to a neighboring cell, which you would do with your conventional mathematical models, but through the distant cell. So think that as a natural mechanism of sensor-actuated technology that we're using in engineering in everyday life. And of course, all of that, the idea and these processes can be modeled, in our opinion, only successfully with non-local models. So what I was trying to convince you in is that, okay, we were talking about biosocial system, and then those slides that you've just seen before were effectively about pattern formations, right? Spatial-temporal patterns in the general way. Now, how you do it mathematically is kind of well-accepted way since Turing time, right? So basically what you do, you can see the reaction diffusion processes. You do not consider just diffusion because, as I said, even if you go to vegetation patterns, right, dispersal becomes very important. Dispersal is a non-local process. So what you basically do, what you start with reaction diffusion processes where you have, in addition to diffusion, you have competing reaction mechanism. And then you may have spatial pattern self-organization. You may have, of course, diffusion-driven instability, which, very often you could analyze from Turing instability. But also you would have non-local interactions. And non-local interactions, when long-range interactions drive spatial aggregation and patchy distribution in such situation. And the example that I'm going to show you effectively would be related to on the border between biosocial dynamics and ecological dynamics, specifically predator-prey type of models. And then, of course, the next step would be that you would like to account for stochastic effects, right? So what is the noise-induced pattern, whether stochasticity may help you to understand better those patterns, and whether even some random effects may induce some patterns which you would not be able to see without accounting of those random effects. And then bifurcation, which is kind of classical thing, but then symmetry breaking. And symmetry breaking, of course, it's another important thing that we have to account for. And then the last example that you would see here is neurodegenerative diseases, right? So again, this is brain study, right? There you have, in many cases, not all, right? You have these diseases associated with abnormal protein aggregation, and that aggregation, fragmentation, and nucleation processes in many cases also are such that you have to take into account those non-locality into account. So, of course, I mean, everyone knows Alzheimer's disease, Parkinson's disease, and many more. Today, I've learned from Stephanie that there is Alexander disease, right, which is another complex things to look at. So this is another group of models that we're going to look at today. But my focus in terms of neurodegenerative diseases, for a number of reasons, is going to be mainly related to Alzheimer's disease. This is something that we know for 120 years, the first time it was described. And after 120 years, we still know very little. We know very little how to deal with that. The thing is that what you have in Alzheimer, you have abnormal structure, right, called plaques and tangles, which is prime suspects in damaging and killing nerve cells, right? Plaques are deposited in protein fragments, which are known as amyloid beta protein, right? And it is built in the spaces between nerve cells, okay? This is specifically related to nerve cells. But then you have also tangles, and those are twisted fibers, and this is another type of protein, which is tau protein, right? And of course, they interact. And then again, misfolded amyloid beta and tau proteins spread in a non-local manner across the brain network, across brain regions, via neural connectivity and extracellular diffusion. Diagnose Alzheimer's is extremely difficult, and that's definitely one of the things that to look at. But this is non-local. But before going to non-locality slide, let me also mention about Alzheimer's. So you may heard that basically the first drug after 20 years break in Alzheimer appeared 2021 or 2022. Right? It was called aducanumab. Now, that drug was targeted this amyloid beta protein aggregation. And you could look at the literature. Scientific community, did not like the idea. because scientific community knew very well that there is very complex interaction between different types of protein. It's not only amyloid beta protein that is important. However, the argument on Food and Drug Administration in the US was, okay, we need to seek at least some relief to those people that suffering from the disease now. And so that's why the drug was introduced. The cost, by the way, of the drug when it was introduced was $40,000 per person a year. So it was enormous money. But as I said, scientific community knew very well that this is not the only cause. The justification was coming only because to relieve the current patients that suffering from this disease. Anyway, so these three examples that I gave you effectively lead us to the idea that, okay, we need to study non-locality. And in terms of biosocial system, this is my first example. So this is interaction between biological and social factors, individual and collective behavior, health and population dynamics. So some of the example, kind of you could see here from all these three areas. And as I said, although I'm not going to talk about that, but definitely COVID, made us looking at these biosocial interactions in a much more serious way compared to how basically we were looking at that before. Because indeed, you cannot describe just biological systems without accounting for social interactions. And this is concerned not only human society, by the way, this is concerned also animal kingdom, as you would see in the next slides. So now mathematics here, underlying mathematics, if you like, or starting point, let me put it in this way, starting point is of course, coupled reaction diffusion systems, right? Where, for example, you could take into account, if you're talking about neurodegenerative diseases, you take into account the evolution of each of these interacting proteins within the same model. And you know quite well that not everything is transported through diffusion. There are many other processes within that that you have to account for. But let's go to ecological dynamics first, and basically it's the system on the border between biosocial dynamics and ecological dynamics, because the first example that you see here is effectively a predator-prey model, right? And what you see here, you see, of course, the diffusion is accounted for, but what we do also here, we account here for the fear. And this is something that also extremely important because this is the first psychological effects that needs to be accounted for when you talk already about the interaction within animal kingdom. You cannot ignore that. And this is something that you have to introduce into the model. And that's what we do. Without this term that you see, an omega, you will see in further slide, is, if you like, indicator of fear that we analyze, Other than that, you see that the model is reasonably kind of standard within the community of predator-prey model. So this accounting for psychological effects, this is something that part of our bigger projects, because as I said, for example, in the COVID dynamics, this is something that you have to account for. But in many other areas, you go to outside of biology and you see that in everyday life as well. So, you see here pretty much the example of the model. However, there is a bigger picture in that our thesis here is that to understand the dynamics, if you analyze interactions within the animal habitats or in the human society, psychological effects cannot be ignored. And the example here, what you see is basically just fear. Now, is fear a local effect? Of course not. Of course, animals feel through the distance, basically, already predator, and they take measure to do that. So if you are going to ignore that fear effect, you would not describe dynamics correctly. And for that reason, the model that you've just seen, we generalize to account for those non-local effects. Mathematically, to analyze those non-local effects, you introduce the convolution terms, like here. So remember that that term that was like completely deterministic, completely local on the previous slide, now become non-local through this convolution operator that you see here. And that convolution operator kind of make a seemingly visually slight difference, but huge difference in terms of the dynamics and in terms of the modeling. That convolution operator is standing here, right? Instead of that just local interaction that you've seen before. So we did this type of model, and this is just a glimpse of what basically you would see. So on the horizontal axis, you see this omega, and this omega is effectively the parameter related to fear, right? And alpha is again for different alpha varying the level of fear. You see that as fear level increases, the prey population declines, which is something that you could affect. However, the rates of declines and many other things are different from what you would do with just standard local model. And what it leads to a reduction in predators. So both type of dynamics, predator and prey, are augmented once you take into account these non-local effects, let alone that I actually forgot to mention that in that model, we also account for the secondary food and refuge as well, which is, I think, is going to be part of on this slide. So M now you see on the horizontal axis, it's effectively prey refuge. And on the left slide, you see prey. On the right slide, you see predator. And the idea here that if you do rigorous analysis here in terms of your temporal system, so the coexisting equilibrium point can be obtained when a certain amount of prey hides in the predator prohibited zone or refuge, if you like, but both populations perform then oscillatory behavior when the prey refuge is below the threshold value. Threshold value is effectively defined through Hopf bifurcation right here. So here is stable range, here would be unstable range. And here, actually the idea also is that what you would see in terms of branching behavior here is that depending on the fear, there will be a shift in terms of these curves. And this is, I think, the last slide related to that. So the Turing curve shifts upwards, effectively, when you increase the range of non-local interaction. And the classical Turing region, which is denoted here as capital R1, also is going to be different. And in fact, it is going to increase in the chance of occurrence of stationary patterns in the population. Of course, you could put that, in a spatial picture, but again, that spatial picture may look trivial for many, although it already gives you indication that non-local interaction results in the formation of larger and more diffuse patches compared to the local interaction. Because the spatial correlations are stronger and spread over a larger area, leading to the emergence broader and connected pattern, as you see on the right part of the section. You cannot expect something more complicated than that, because effectively the model that you saw is effectively one-dimensional spatial model, right? The spatial temporal model. So that's what you get. However, you already getting insight based on that. Now, you could actually see a little bit more. So what we will analyze here, it's non-local predator-prey model. However, not like one-dimensional, but actually in a habitat, in more realistic habitat. However, other parts of what you saw before is effectively this convolution that are related to accounting for non-local effects are here. And of course, this convolution operator are kind of becoming two-dimensional, a little bit more complicated. The other thing that the kernel that you would see here, right, which enter this convolution operator, this sigma will be a characteristic of the non-local range interaction. So the plots that you're going to see later on the slides will be plotted with respect to this sigma. So how you effectively change or account for non-local interaction will influence the dynamics that you will be looking at. Okay, so that's the idea. And of course, the range of the patterns that you see in this particular case, and again, you're just looking at Turing patterns for the local model here, of course, much richer compared to what you see on the previous slide. Even in this case, of course, you already see that local model already introduced non-homogeneous stationary Turing patterns. But now the goal is go beyond that. Now the goal is to go to account for non-local interactions, because what you see here already is rich behavior. However, this rich behavior could be described as conventional Turing patterns, and this can be done with local model. So what basically non-locality, what does convolution operator bringing us to the table? Well, first of all, the simplest case to analyze is hexagonal patterns, right? And on the left part, you will see effectively this is weak nonlinear analysis of the model that we introduced. And that's why the y-axis is given in terms of the amplitude. But you could describe pretty much all patterns, and they're given on the left part of the picture, depending on the certain parameter, that capital phi, for example, here, 0, pi, and so on. That's what you see before. But stability of these hexagonal patterns for the local model is basically shown on the left, whereas on the right, you see that the effect of non-local parameter on the Turing bifurcation curves. So basically, Turing bifurcation curve will be shifted. So it's not that the dynamics is going to be the same. Once the Turing bifurcation curve is shifted, you will have much richer and different dynamics compared to what you could describe with just local interaction model. So what you see here as a title, non-local interaction enables effectively Turing patterns across much larger set of the parameter values. And what you see, non-local model shows non-homogeneous stationary patterns for a wider range of parameter value compared to the local model. And you see different patterns here, right? You may recall the beginning of my talk, right? When I started with vegetation model, then I moved to animal kingdom model. Those patterns may remind you what you see here on this slide, right? And this is due to the fact that we account for these non-local effects. You see here the same kind of terminology, hot spot to cold spot, A and C, and then stripe to cold spots, which is used also even for the model in ecology for the vegetation. Now, of course, you have to see what randomness, or what we call randomness, right? The facts that we may not know within these dynamics may bring to the picture. And this is something that you could see we actually published very recent this year paper, where we started with the extension of very well known, right, Rosenway Alzheimer model, which incorporate density dependent death rate in predator with the following dynamics. Right. So this simple model was augmented to account for spatial temporal dynamics with perturbing parameter R and M, as you see below, with corresponding mathematical expectation for the stochastic disturbances. So of course, this is simply a model compared to what you see before, but what you could do is that you could effectively analyze these confidence ellipses, right, for this stochastic system that you're trying to analyze around the stable equilibrium point. And these stochastic states are distributed around the stable deterministic attractor. However, the size of these confidence ellipses will increase when the stochastic disturbances will be increasing. But it's not the whole story, right? It's not the whole story, because effectively what you see for small noise intensity, you still will be doing okay, even with your deterministic model. And the reason for that is because stochastic states lie well inside the corresponding confidence ellipse. However, they do not always stay within this confidence ellipse for higher noise intensity. So that's definitely a challenge. So that's the picture effectively when everything is okay. When disturbances, here's the sigma actually is characterized as disturbance level. So when disturbances are small, everything stands fine. Confidence band expands with noise intensity, yes, but stochastic trajectories stay well inside the band for this noise intensity. What becomes challenging when the level of this noise is increasing. So there is a bad news and there is a good news. Bad news, of course, the things becoming much more complicated. So the dynamics becoming much more rich. However, what is important here to find out is that the noise-induced spatial temporal model shows self-organization patches in the wider range of diffusion parameter value and produces different patterns depending on the level of that environmental variability, if you like, with which you model those random or stochastic effects. So the bad news, yes, things become more complicated. However, the good news is that this is something that brings us to the patterns otherwise you would not see. So what's next? Next is basically the model that we did for which also available at the PLOS one. Is related, so basically this part of the talk was related to biosocial dynamics, if you like. However, on the border also with ecological model, because, how do you classify predator-prey model, right? It's kind of biosocial dynamics, but also directly related to ecology. But now it's ecosystem, if you like, model. And what you see here, again, mathematically underlying system, it's coupled reaction diffusion system, right? Depends what you put in the reaction, what you could do in the reaction. And what you see here, it's so-called semi-arid environment, where you write the system with respect to two things. One is the water density in certain area, right? And then the other variable, V, which is responsible for plant density, right? And what we did, we took this and the connection here. So what we introduced also, we introduced here herbivore, right? This is animals who actually feed on plants, right? In this ecosystem. And we introduced Holland type 2 functional response, right? Holland also was Canadian. And that system has been generalized to, as you see, to account for this herbivore dynamics. So you have three coupled reaction diffusion equations, and you analyze this system again in terms of pattern formation and in terms of effect of water flow on this stationary pattern. And the non-homogeneous stationary pattern move in the presence of water diffusion and downhill water flow, which you would not see, again, with the original model that I put on the previous slide. So this is something that new you analyze and you see. Now, the thing is, if you go to this, to deeper analysis of these non-homogeneous, non-stationary solutions for the plant, depending on the parameter value, this temporal model predicts catastrophic shift for the parameter value in the Hopf unstable domain. You may know this terminology, catastrophic shift. I think was introduced by Martin Schaffer in Nature paper like 2001. And since then, but the good thing, again, there is always bad and good news. The good thing is that small diffusion parameter value of the water can revive plants and herbivores from extinction. So the next step, if you like, behind this would be probably to introduce some control problem, how to choose these parameters in such a way that ecological system will be preserved. So that's an example from ecology. And now I'm going to, as long as I could click, I don't know, okay. Yeah, so now I'm going to the third class of problems that I'm going to talk about is effectively non-local model in the dynamics of neurodegenerative diseases. So the first paper that we published on that was in Scientific Report a few years back. What you see here, you may recall what I was mentioning about aducanumab drug that has been introduced a few years back, which was targeting these amyloid beta proteins, right? And I told you, and this is not only us, but all scientific community understood that very well. That drug, goes, in some way contradictory to what scientific community already knew. We have very complicated interaction between not only amyloid beta, but also tau protein. And this is something that has to be taken into account. This is non-local model. Non-locality here is modeled with again, with convolution operators. You already have seen how those convolution operators are introduced, but you see these convolution operators basically everywhere here. So the notations here is, you could view that U and V are effectively healthy protein. So U are related to healthy amyloid beta protein, V are related to healthy tau protein. And then with tilde, the second and the fourth equation, it's effectively already damaged protein, already unhealthy or toxic protein, as we call it. And then you have additional equation, again, non-local equation, where you look at the evolution of the non-local brain damage. Again, you see the very last equation, again, contains this convolution operator. So you have go beyond like standard framework of PDE, of course, well beyond ODE. You go to integral differential equations for the reason to introduce those non-local effects. Now, the question is how valid your result would be if you write your system of an even integral differential equation with respect to the realistic data, right? The realistic data that we've been using was effectively based on the human connectome project, right? However, to use the data, you need to move from that, spatial temporal model that you are using to the model on the network, because that's how the data is given for you in that connectome project. So you have to approximate, if you like, your Laplacian and all other terms, your convolution term on brain networks. And, we did that part, and you will see basically, several similar ideas. And then you have basically the system like this. Of course, the number of equations is increasing. However, you kind of move from PDEs, no, not exactly PDE, integral differential equation, right? But from partial derivative in time, you kind of move to ordinary derivative on the graph Laplacian here, on the network, in other words, right? Where all these kind of, we don't have time to do that, but you could see clearly that all these non-local effects are preserved here on these graph networks, right? And that's the definition of this convolution operator on the networks. Depends on the weight vector, the number of elements in the network you are taking, and so on and so forth. So you have to be kind of constructive doing that. Now, what you see here, you see all these four major parameters, if you like. So the top and the bottom, what you see in solid line, right? Let's put that here. I don't know how to do that, but it will go. So top and bottom solid line, it's effectively related to healthy protein. And you see this is a dash dot line, this one and this one, it's toxic amyloid beta and tau protein. So first you look, of course, at local model, what you could do with the local model, and what you see here is the local model shows delay in disease propagation, and the toxic load decreases with the increase of the reaction time. So the effect of the non-local parameter, this is something that you would see in a second. This is related to local model, this picture, because what you see here that the actual local model shows almost homogeneous distribution of toxic loads in the brain connectome for primary tauopathy. Primary tauopathy is basically here, the major source of your neurodegeneration is coming from tau protein. Okay, it's not mixed, which more related to Alzheimer's disease. This is more related to these type of effects. Now, however, you see a different picture when you account for this non-local interaction. So the density of toxic protein on a node depends on its degree. And the non-local model predicts it's better than local model compared to the data that you would have within human connectome. And what is very important is non-local parameter slows down the disease progression. So, some claims that people have been making based on the local model are not completely true if you account for that fact. Of course, the picture itself, in terms of the evolution, what you see on the left, it's kind of, larger scale picture. What you see on the right, it's kind of more zoom picture. And this is node-wise toxic tau protein distribution based on the non-local model. And, this range of different curves that you see on the right corresponds to different brain regions. And we will see on other slides which brain regions we actually accounted for. So again, emphasizing that the local model shows non-homogeneous distribution and different propagation profiles of this toxic tau protein in the brain connectome, including not only, as I mentioned, primary tauopathy, but also mixed tauopathy, which is more relevant to Alzheimer's disease. Now, this is something that I spent my morning was fascinated what Stephanie is doing related to astrocytes. But as I told her, and I repeated for the entire audience, when I see astrocytes, it's mind-blowing. And the reason for mind-blowing is that topology of this structure is so complex, and they do so many things, many good things, but also many bad things. And here some of them, highlighted, because now we going, even though we called it neurodegenerative diseases, now we go to different type of cells. Those are glial cells. They are not nervous cells. However, they play a crucial role, and a bunch of things are highlighted here. So, they first of all, they're in contact because of their very complex topology. They're in contact with thousands of neurons. And these cells play an important role in the entire nervous system for both for us human, but also for mammals, genetically. So positive things is astrocytes support metabolically to ensure their optimal functioning. And without this help, neurons will not be communicating. But on the negative, so they maintain effectively, here you see in this line, they maintain a healthy balance in the brain by clearing this amyloid beta plaques. So this is positive thing. But look next. However, if they are overactivated, so these astrocytes release quite harmful things in the presence of amyloid beta and also react to pro-inflammatory cytokinase and further increase in the production of amyloid beta. So it's kind of double-sided sword. On the one hand, they have positive functions, but on the other side, they have negative functions. And so what we do, we use also, I've heard that today that Stephanie also using Smolhovsky model. Why to use Smolhovsky model in this field? Well, Smolhovsky, Marian Smolhovsky was the first who actually, said that, first of all, if something is dependent on the size, by definition, it's already, you have to use non-local model. But he went beyond that. He said that, this thing's aggregation depends not only on the size of the particles, but also on the distribution of the particle. So that's the reason why we believe that, again, you use, as you see here, a reaction diffusion model. So you have diffusion operator, but then you have a bunch of other terms. You have nucleation, you have aggregation, you have fragmentation that you have to account for. So, and we need that, which I don't think I have much time to go, but you go through this process step by step. So you saw those terms, you go through nucleation, then you go through aggregation, and you go through fragmentation. And then what do? You have to account for connectomic data. And to account for connectomic data, what do you need to do? You have to reduce your reaction diffusion equation on a connectomic network. That's the only way how you could account for this type of behavior. And that's what you see. This is basically the structure of that system where you see healthy protein, amyloid beta protein, healthy tau protein, toxic amyloid beta, toxic tau protein, all together as coupled, however you see that on network, and that's where you could use connectomic data, based on this model. And, this is very simple thing, but first size-dependent clearance and fragmentation. So what you see on the middle point, on the middle plot here, which seemingly, deceptively simple, but the thing is that middle plot shows that the astrocytes have the biggest influence on amyloid beta during the seeding stage in the absence of fragmentation. Okay, so there is much more behind that, but I would not have time to cover everything. Remember, one of the plots that you saw for different brain regions, right? This is another example of that. So basically what we're considering here, limbic, frontal, basal ganglia, parietal, temporal, occipital, and brain stem itself. So what you see, what I listed here is basically from bottom to top, different part. And of course, some of the nodes do not follow the same spreading behavior due to the fact that they are disconnected from other nodes. However, the spreading behavior of toxic protein amyloid beta of all regions is pretty much the same. So, and again, remember that all that has been done within those non-local models. Now, integral differential equations is not only, even though you kind of reduce those integral differential equations on the network, still it's not the only way to introduce non-locality in your mathematical or statistical model. The other way is to go through fractional calculus, right? And so basically you introduce fractional derivative, and this is something very recent. Again, we published paper this year on that. But you use Caputo fractional derivative. The thing is that what you see here, alpha, right? When alpha is alpha equals 1 is basically more or less standard derivatives, right? And then you decrease this alpha, and this reflects fractionality of your model and also how you account for those non-local effects. I don't have that much time here, just pointing out for me. But what you do here, again, you do reduction to the network, to connectome network. And then what is interesting here to highlight is the higher memory takes more time to distribute the overall toxic loads. And again, we cannot kind of stay within this too long, but a few things. Remember, I mentioned alpha. Alpha 1 is basically standard calculus, right? So standard integral differential equation. Alpha reducing, you introduce fractionality. The smaller alpha is effectively the more non-local effects you can catch. And again, this depends on the brain region. You see kind of different plot, more interesting dynamics. And here more color picture, if you like. Memory effects on brain connectomes. What is interesting, I probably should also highlight, in biological sciences, not only in biological, in physics as well, except of quantum mechanics, we're always talking about Markovian behavior. Now, this is the area where you have to be very careful with this assumption, in our opinion. Sometimes this dynamics is much richer than Markovian dynamics, and this is directly related to the accounting, this memory effects that we are talking about here. In fact, the original paper that we publish is starting from non-Markovian dynamics. Now, the very last part for a few minutes is non-equilibrium dynamics. And many of you kind of know that non-equilibrium dynamics is much richer, but also much more complicated to analyze mathematically. Here, the major groups, if you like, of models and problems that we were looking for is effectively calcium, related to calcium, right? Interaction of calcium with other stuff. And calcium oscillation, exciticity, calcium-induced synaptic loss, stochastic calcium spike, but also related to Alzheimer is amyloid beta wavefront propagation, which is important. And then you go to network effects, which is also pretty rich area. I cannot go too far into details, but a few things here that if you use even this simple model of interaction amyloid beta with intracellular calcium behavior, you extend this to stochastic effects. By the way, the second term here in the original model, it's effectively the model to which contributed, the person after which this institute was named, right? She did her PhD in 1911, then she moved to Germany, in Mihalis Menten dynamics is effectively the second term that you see here in this model. So we extended that to the account for random effect, for stochastic effects, and then again you do this procedure that I was describing, I guess, before. So and then you study the stochastic dynamics with this confidence ellipses. However, it's not only extension when you induce these perturbation effects. What effectively is happening, the trajectory for the deterministic system do not necessarily cross separatrix, which you see basically in this case, which is effectively related to Hopf bifurcation here. And yeah, we don't have much time. This is kind of, three-dimensional picture related to that. And again, if someone is interested, this paper has been published, so we can chat more about that. Right. The very last thing which I would like to mention is we do also, because I do know that some students here from statistics department, right? We do some part related to machine learning and artificial intelligence, but this is in the context of optimal control problem. So what you effectively do when you do drug administration for neurodegenerative diseases, you would like to see how the drug and for how long the drug would affect the patient and how long it will be good and after that. So that's basically the idea. And effectively what we do here, again, I don't have time to stay on that, but you, as I said, you formulate that as a certain control problem, right? You develop certain algorithm, but what is important, of course, Bayesian statistics is extremely important. However, in our opinion, Bayesian statistics does not give you the full picture of what you need to do for this type of problems. You need to combine basically what Bayesian statistics does for you. It allows you to evaluate, if you like, quantify the uncertainty. But then also what you need to do, you need to understand the complex dynamics behind that, based on the data that you have. And so what we effectively do here, we combine Bayesian statistics with physics-informed neural networks, which do the two parts for you. And, of course, you need to study like training data and things like that. Here we did just 3 hidden layers for this type of problems. But, you could definitely see that the, training with the increase in training numbers, the MSE loss function will be decreasing basically for pretty much. This is my pretty much last slide before. So a few things that I would like to mention here. So this is not something that I'm telling in my talk for the first time, of course. So one of the first papers that very clearly emphasized that brain dynamics are known to be non-equilibrium. At least, in Pro paper and other 2021, you could clearly, it has been stated. Now, neural systems in general and brain networks models in particular are being studied based on 2 main approaches. One that you've seen, I guess, quite prominently here, which is related biologically inspired approaches, which effectively you use data, for example, from connectome type models, then you do discretization. And then Bayesian approach. What I would like to mention here, and that's the reason why we use combination of Bayesian approach and physics-informed neural network, is because, again, you could look at the literature. Bayesian approaches challenge empirically so many times this experimental fact, specifically in the decision-making, and providing examples of breakthrough of the classical framework of cognitive sciences. They will not work alone in all situations, because nowadays with the income of artificial intelligence, right, everyone is saying Bayesian approach. No, it's not the remedy for everything. In some cases, you need to go more. What the very final thing, because I know that there are, mostly graduate students here, if you're interested in that area, look at that, because effectively for brain, right, what is very important also for Alzheimer's disease, right, working memory. Working memory, it's effectively initial, inset of this Alzheimer's disease. That's how you could say. Now, how to model working memory? Working memory is modeled through the learning. Learning is non-equilibrium dynamics. It's not equilibrium dynamics. Therefore, all kind of these things. And our motivation here, of course, is larger than that. So as you see, there are some references related to that, because then you go from here, you go to decision making, right? And then you go to biosocial system for the human society, and you move on. Without that, I don't know. I mean, I think we could stay at the same stage of the development of science if we still just look at the Bayesian approaches for all problems that we need to address. Okay, so that's pretty much all what I would like to share with you. This is the summary, but I guess I was trying to highlight as we go the non-local models for all of these three big groups, right? Biosocial dynamics, ecological models, and neurodegenerative diseases. And the last part of my talk was related to non-equilibrium dynamics that can occur in brain, including higher noise intensity, and then the combination of these spin and Bayesian approaches. So yeah, of course, I mean, there are many things to do. Some of them I highlighted here. Of course, kernel that you use in your convolution terms, this is something, even though you use non-local model, no one can tell you which kernel to use, right? And of course, Gaussian kernel is something that you start from, but whether this is the best choice depends on the data you have. So this is one thing, and there are a bunch of kind of other stuff here, which is kind of exciting, and I would be happy to talk to anyone. Not all, but most of the things have been published. The thing is, we also have a review on non-local model in Physics of Life reviews, published just recently. And yeah, if you are interested in any particular case, so this one that I was mentioning, that not everything is Markovian dynamics, including here, very recent paper as well. And I guess on that, I just would like to thank you again for invitation, for the opportunity to talk with you. And if you have any questions, so I would be very happy to respond now or later. Thank you.